Hello, I'm Jingyu Zhao from the UNC Chapel Hill. I'm the fifth year graduate student, and I'm a awardee of the PhRMA Foundation Predoctoral Fellowship in Drug Discovery. I think my interest of the science started at the moments that I realized the science can really change people's life. I remember it's a seminar in my master training. One of the speakers are from the stem cells and tissue engineering lab. And their first slides just show an image that I will never forget. It's a mouse has a human ear structure on its back. And they introduce a lot of things about how they use the biomaterials and biological techniques to help rebuild the structure. And they also tell a lot of story about that how they help the people who experience severe trauma, such as a car accident, to reconstruct their facial or cranial structure. So it's the first time that I feel like how the science could be. So it's not just the experiment or the data, it's actually the real story that how the science can improve the people's qualities of life. So finally, I joined that lab. So that's the start of my research career. So my research focuses on the in vivo CAR T. CAR T therapy is a very successful immunotherapy that they engineer the patient's own T cells to express the artificial receptor we call CAR to help kill and recognize the cancer cells. The current CAR T therapy is highly expensive, which was around 500,000. And if we considering the treatment of the side effect and the hospitalization, the total expense would over $1 million. So why the CAR T therapy is so expensive? That's because it's special logistics. The current CAR T therapy need to collect the patient's own T cells from the blood and transport the blood to the central manufacturing facility. And their blood need to be isolated and activated and finally infused back to the patients. The entire process may take 2 to 3 weeks. And in many ways, the therapy is more like a service rather than a traditional drug. So in my study, we proposed an innovative method called in vivo CAR T. We are aiming to design some vectors that can directly engineer the patient T cells in the bloodstream. In that way, we believe we could provide the off-the-shelf products and can get the patient get their treatment just in a single day. More importantly, we believe the current CAR T therapy, the T cells would undergo the extended ex vivo manipulation, which may impair the functionality of those T cells. So in our in vivo CAR T strategies, we just protect the T cells from the ex vivo culturing and activation and expansion. So probably we would provide a more efficient method. So I think one of the very important moment during my research journey, I remember that was the first year of my PhD study, and during my rotation, I was doing molecular cloning experiment, but I found no matter what I tried, it just didn't work. Another 2 students rotating with me, they got beautiful data, they got everything moved like smoothly, and the PI are happy about that. But just for me, I'm the only one that tried everything, but everything failed. So I was super discouraged and I feel a lot of peer pressure. I have a run-of-the-run discussion with my PI and talk to the senior student, to the postdocs, and read a lot of papers, try to find out what's wrong with that. But fortunately, I finally figured out what happened and I fixed it. But I think compared to today's experience, that's just a very small moment. But I think that's the first lesson that to make me feel like how the scientific research would be. The peer pressures, comparison, the setbacks, but the only thing that we would do is just keep going. I think my career goal is to become an independent PI, and I hope to run my own lab. I'm excited to work with the talented people, collaborate as a team, and I will continue focusing on the drug discovery. And from the pharmaceutical industry perspective, I would like to work with some project related to the translational medicine, and I hope my research can really enter into the clinical trial and benefit more people.